I did my research with the Evason lab with the Department of Pathology of the Huntsman Cancer Institute. And this is my research on getansrin and retransferrin drug treatments for hepatocellular carcinoma, as well as HDR2B overexpression transgenic zebrafish models. Kind of a mouthful. So hepatocellular carcinoma, we'll call it HCC from here on. It's the fifth most common cancer in the world and the third leading cause of cancer-related mortality. Um, it's the most common type of primary liver cancer, making up 80% of all liver cancers. Um, and a big issue with this particular type of cancer is that only 10 to 20% of HCC can be removed surgically. And even when they can be removed surgically, there is a recurrence rate of 80% at 5 years, which demands more chemotherapeutic options for the treatment, which is what this research is focused around. The median survival rate following diagnosis is 6 to 20 months. The 2-year survival rate is less than 50%, and the 5-year is only 10%. So, pretty awful statistics. Another weird factoid is that the ratio of HCC in males to females is 4.5 to 1. It's much more common in males than females, and nobody's sure why yet. There's evidence of an androgen, perhaps, um, causing more proliferation. But this cancer is not common in children. In the lower left, you can see a map distribution of HCC throughout the world. Bottom right, you can see the progression from a normal liver to a cirrhotic liver and one with HCC. This shows that HCC usually goes in hand with overall liver damage, which is called cirrhosis. This can be caused by toxins, alcohol, bad diet, and also very commonly hepatitis. In this lab and in this research, we use the zebrafish animal model to study human diseases, in this case, HCC. An advantage to this is that it's easy and cost effective to house and maintain. Um, in comparison to mice, which are more genetically similar, being mammals, zebrafish are vertebrates, but they are fish. They require much less space and are way cheaper to maintain. They also breed readily and have hundreds of eggs at a time. It increases your sample size for whatever drug or toxicity experiment you are doing. And they're also very easy to genetically manipulate. And a key reason people use zebrafish is that the embryos and larvae are translucent. You can see right through, see what's happening, see the development. And so for our research, where we look at liver size, we are able to see the liver pretty late into the stages of development. It's not until they mature pretty far that they actually get that pigment in the skin that doesn't allow you to see anymore the all the internal organs. And most importantly, 70% of human genes are found in zebrafish, and this makes our research valid in humans. And there's also an expansive scientific community. There's a ton of literature on breeding techniques, developing transgenic and mutated models, and at this point, since they are so popular, almost the entire genome has been sequenced. So this image on the left is injecting a zebrafish embryo with a DNA or RNA construct. This is how we produce a lot of our transgenic fish. Just directly inject into the one-cell stage, as you can see on the left. And on the right is a state-of-the-art zebrafish facility. This isn't the one at the HCI or the ZAR, the Centralized Zebrafish Animal Resource Facility. We call it the ZAR, but it looks almost identical to that. And this just shows you the power of these facilities and the zebrafish models, that those rows hold thousands and thousands and thousands of zebrafish. The beta-catenin model of HCC is what we use in our lab. Our lab has pioneered this. 20 to 40% of HCCs have mutations in genes encoding for activated beta-catenin. And what beta-catenin is a protein that normally contributes to cellular development as well as stem cell and organ renewal. It acts as a transcription factor and plays a role in WNT signaling. And also 33% of beta-catenin transgenic fish we developed in our lab developed HCC at 2 months post-fertilization. And that number increased to 78% at 6 months and 80% at 12 months, which shows you how powerful this signal cascade is in the development of hepatocellular carcinoma. And on the bottom left, you can see how the construct actually works. The beta-catenin is expressed exclusively in the liver, and we have green fluorescent protein expressed specifically in the eye, which allows us for easy identification. And on the right, you can see a liver size experiment, which shows that our transgenic beta-catenin fish have a much larger liver, which is a good predictor for whether the fish will eventually develop HCC. And these liver size experiments are the same things we will do later on. Serotonin receptor HGR2B. So you probably know serotonin is a very common neurotransmitter that makes you feel good. And if you don't have enough, you maybe become depressed. And if you have too much, you're jittery. But actually, most of serotonin signaling is done outside of the brain. It's called peripheral serotonin. And the liver in particular uses serotonin signaling in regeneration. After everyone knows that the liver is great at regenerating. Like you can damage it, and if you be healthy and drink a lot of water for a while, it will regenerate. 29% of human tumors tested positive for the HGR2B receptor, and 35% of patients with HCC. And this is also associated with an increased proliferation index. HGR2B is suggested to be the mediator of survival. It leads to sustained phosphorylation of downstream targets mTOR, P70, S6K. All of these pathways facilitate the survival and inhibit autophagy. And the inhibition of 5 HT2B receptors leads to cell death of HCC cells in vitro and decreased tumor growth in mice. So this is what we're trying to experiment with in our zebrafish models. And HGR2B is a serotonin receptor just in the family of all the HT serotonin receptors. HT2A and 2B have shown to be very influential on the progression of liver cancer. And HGR2B seems to have more of an effect, which is why we have chosen it to be overexpressed in a fish. And the construct we use is actually identical to the beta-catenin construct that is renowned, except instead of beta-catenin being exclusively expressed in the liver, it is now HGR2B, which is overexpressed in the liver. Okay, the two drugs we used are catanserin and ritanserin. Very similar sounding and very similar properties and activity. A big benefit is that they are both already FDA approved, which would skip many costly steps in the approval process if our treatments are found to be effective. They are both 5-HT2 receptor antagonists. They both have high affinity and are non-selective. Non-selective meaning the HT2A, HT2B, HT2C, HT2 all receptors will be antagonized by these drugs. The key difference, though, is their activity with VMAT. But quickly, I'll show you the antagonistic properties of our catanserin and ritanserin. As you can see, if you add these drug treatments, you're going to block the signal cascade. And VMAT is a transporter that serves to shuttle serotonin into vesicles to prevent degradation and prepare for release in serotonin signaling within platelets. Most of all peripheral serotonin is stored in platelets. So using both catanserin and ritanserin will allow us to more clearly see if any change in liver size is caused by blocking the HGR2B receptor or doing that, but also inhibiting VMAT, as in catanserin. So it's very important. So now to actually some data that we collected. We carried out 2 catanserin experiments this semester. In the first experiment, we did not see any significance between control and treatment groups. All these techniques are very specialized and require a lot of skill. So it wasn't a huge surprise that we saw no significance. If you look closely, you can see there is an overall decrease in liver size of the treatment groups, but it was not below our p-value of 0.05. Yeah, excuse me. In experiment 2, you can see that we actually did achieve significance in our treatment groups, which is awesome, which is what we wanted. And so this has shown that with ketanserin, you do have a decreased liver size in both the beta-catenin model and in just normal zebrafish wild type. Ritanserin. We only carried out one ritanserin experiment throughout the semester. In it, we did not see any significance. You can also, if you look very closely, you can see a slight decrease, but not below our p-value once again. This experiment we have repeated. We have collected the data. We now just need to analyze it for a second experiment, but so far, no significance. Our ACR2B overexpression fish, same liver size experiments here. In our first clutch, we saw no significance. It was a much smaller clutch. And then our second clutch was much larger. And we still saw no significance. Why? Well, there are many possibilities, but my best guess is, cancer, as we know, is not just one mutation. It is many mutations. Proto-oncogenes mutating to become oncogenes. Tumor suppressor genes mutating to where they no longer carry out their proper functions. Mutations that allow the tumor to become metastatic and spread into the bloodstream. ACR2B expression alone appears not to be sufficient to throw the liver cells out of their normal cell cycle rails, especially not 6 days into development, which is where we measure liver size. This brings me to the next part of my research, which is crossing ACR2B overexpression fish with beta-catenin fish. So we will cross these two transgenic lines and see if liver growth is amplified by this or has a multiplicative effect. And from there, we'll be able to tell how much, if any, ACR2B contributes to liver proliferation. Upcoming work. So my research took a big hit with the coronavirus pandemic. The HCI suspended all non-essential employees as there are immunocompromised cancer patients in the same building. So I really could not do much. But in May, I hope to start working on my project again and seeing the influence of ACR2B overexpression combined with beta-catenin on the development of liver cancer. We also repeated a ritanserin experiment. All that data has been collected. We just need to analyze it. So I'm really hopeful to see the data there. And another direction we plan on taking our research is cross overexpression fish for ACR2B with MAOA knockout fish. MAOA, sorry for another acronym, is monoamine oxidase. It's an enzyme that breaks down most serotonin in the liver, as well as dopamine and norepinephrine. A cool fact is MAO inhibitors are actually used as antidepressants. Knocking it out increases all the serotonin in the body. So maybe if with this mutation as well as ACR2B overexpression, this will be sufficient to cause a significant increase in proliferation and liver size. Would quickly like to acknowledge a few people. Kim Evason, MD, PhD. She is my mentor and principal investigator of my lab. Liam O'Brien, Richard Smith, Alexis Ross. These are lab colleagues that taught me many, many techniques and helped me along the way. And a special thanks to UROP for funding my research. Thank you, everyone. I really appreciate it.